Okay, all right, everyone. Welcome to this presentation. And today we'll be speaking with Adam D'Angelo of the Pawpaw Project. And my name is Chris O'Mannix. I'm a volunteer board member of NAFEX, or North American Fruit Explorers. I'm the vice president. And before I introduce Adam, I want to go over a few housekeeping items and then talk a little bit about NAFEX as we allow people to filter in. So this is a webinar, and you've probably heard this spiel already if you've been on any of the other talks. So this is a webinar, and unlike other Zoom meetings, participants' audio and video features are automatically disabled. You'll notice that in this webinar, you can communicate with other folks. Just keep it civil and respectful. That's all I ask, and I think it'll go great. Feel free to drop links that you think are relevant, ideas or comments down in the chat. I would love for people to drop right now if they want to where they're from, so we can get a sense of where everybody's from here. We got about 31 people on currently. So we encourage you to ask questions. And so the chat feature is not the best place to ask the question. It can sometimes get lost. So ask there. You'll see a Q&A menu tab. And so ask the topical questions there. Just I ask that you keep them. Don't pepper too many questions, and also kind of formulate your thought. Don't add if you made an error in there or something, just don't add a bunch of other words in there. Just kind of clog stuff up. And then this webinar is being recorded, and it'll be made available to you and our fellow members at nafex.org. And any of the bonus resources, presentations, or other files that presenters want to offer will be made there. After a year, it will go over to our NAFEX TV channel. And so a few words about NAFEX for people that have been joining here for the first time. We were founded in 1967 as a network of individuals throughout the United States and Canada, devoted to the discovery, cultivation, and appreciation of superior varieties of fruits and nuts. NAFEX members here share ideas, information, experience, and fruit propagating material via our website, our social media channels, mostly Facebook, our fruit-specific interest group meetings, and the annual conferences, just like this one, whether in person or online. And one of these days, hopefully next year, we will be going back to online, probably do a hybrid model. As a paid NAFEX member, you will get 4 editions of the Pomona Journal each year as well. And that allows you also to search back through 50 years of Pomonas in our digital library. There's a huge amount of information there, and I recommend if you care about fruit history to go and look at what's been going on over all these years. And then if you haven't, for some reason, visited the website, please go to nafex.org. So it is my pleasure to introduce our speaker today, Adam D'Angelo, and I will turn it over to him in 1 second. But I do want to acknowledge our sponsors as well. So we have our sponsors as One Green World, Copenhagen Farms, and Southern Cultured Orchards and Nursery. One Green World does have a code, and if you want to get 10% off, use the code NAFEX, all capital letters, to receive 10% off at checkout. So I'm going to stop my screen share here, and I'm going to read the bio for Adam. So Adam today, he'll be talking about sustainable agriculture, the future of it, and how Pawpaw is interwoven into that narrative. Project Pawpaw, who he's the founder of and the executive director, is a collaborative effort to develop varieties of pawpaw, Asimina triloba, that better meet the needs of both farmers and consumers. Project Pawpaw aims to develop new cultivars of pawpaws which taste great, grow well, and have longer shelf lives. Their work is entirely crowd-funded, and I think that's one cool thing about this model. All their funding comes from the sale of products through their online store and at in-person events. This funding structure gives them the agility to explore new areas of scientific inquiry while keeping them efficient. And so join us today to learn more about that project and also Adam's other breeding efforts. So, Adam, are you ready?
Well, hi folks. I'm really excited to be here. I've been a NAFEX member for a long time actually. I've just been lurking under a different email than I currently have, but it's been really cool to get to come to these. And one of the greatest things about this is sort of the network it enables people to build and the things it allows people to do. Because a lot of this work has stemmed from connections I've made through NAFEX and NNGA and other organizations like that. And plant breeding is particularly of these lesser known, species is so important. It's about person or relationship building between people, and this has just been a fantastic jumping off point. All right, so like we said, my name is Adam D'Angelo. I'm the founder of Project Pawpaw. It's a crowdfunded pawpaw research and breeding program. And I'm really excited to tell you about it, about some of the work we're doing, sort of about the origin of the organization, about what we've done, where we're heading, and then also at the end we'll give you a glimpse into the pawpaw industry in the US, but also the pawpaw industry abroad, because these fruits, which are native to the US, are actually finding homes on orchards and farms around the world. And I got to take a trip to Europe this just a few weeks ago and tour some of those orchards, and I'm really excited to share what it looks like across the pond. So let's start with an introduction. So, who is this guy? My name is Adam. I'm Adam D'Angelo. I already said I'm the founder of Project Pawpaw. That's my side project. My full-time job is with the Savannah Institute, which is a agricultural nonprofit that's promoting agroforestry and specifically agroforestry in the Midwest. I'm the breeding operations manager, so I'll describe what I do there, but pretty much we have a bunch of plant breeding programs, very similar to what I'll describe today about pawpaws, but for a few other species, and we're trying to make new varieties that are better adapted for these systems to help farmers have alternative crops that they can grow to for a whole host of reasons, both economic and environmental. And I'm a lifelong fruit and nut enthusiast. I've been planting and growing weird fruit trees since I was 11. So really, it's been a very fulfilling lifelong hobby so far. And I'm so lucky I have a lot of more time to continue to do it. So this is these are some slides about my time at Savannah Institute and what we do. So we have about six active crop breeding programs at Savannah Institute. So we're currently actively breeding hazelnut, hybrid hazelnuts. So we're breeding a mix of European and American hazelnuts for the Midwest. We're working on Chinese chestnut. We have big programs in blackcurrant and elderberry, which are very exciting. We planted out thousands and thousands of bushes last year. I think 3,500 blackcurrants and 3,500 elderberry, and we're going to be getting to go through and make selections in the years to come. And these are one of the nice crops because they are so quick compared to some of the other tree crops that we work on. So we'll really be able to see some rapid progress and deliver some new varieties to people. So there's probably going to be more talks about this at some point, so hopefully you can all tune in when that comes around. And in the meantime, you can check out the Savannah Institute's website, and that's Savannah, Savanna with no H. It's named after the land ecotype, not the place in Georgia. And we also have an American persimmon breeding program, which is very cool. Mostly going for cold hardiness. We have about 50,000 seedlings in a nursery in the middle of Wisconsin, and we're letting them freeze to the ground and planting the most cold hardy ones out into orchards to make sure that they have things like great fruit quality and good agronomic traits. And we're also looking at making seed sterile mulberries and a few other aggressive tree crops that have a lot of great characteristics, but just need to be wrangled a little bit. So that's Savannah Institute, my educational background, and the reason why I've been able to do some of these things and why I'm qualified to talk about it. These are I have degree in plant biology and agriculture and food systems from Rutgers University, and I went to grad school for plant breeding and plant genetics at University of Wisconsin. Back at Rutgers, I worked in the Rutgers hazelnut breeding program. So that was my introduction into the world of tree crop breeding, because I had been interested and enthusiastic about them for a very long time, but I never had really gotten to do the science side of things until I became a undergraduate hourly worker in this lab. So this is a very famous program. It's under Dr. Tom Molnar, and a lot of you listening to this probably have already heard this spiel and know about this. But uh, essentially, the too long didn't read is that the European hazelnut is a wonderful plant, makes high quality hazelnuts that are the idiot type of what we eat and consume as filberts and hazelnuts today. The issue is that when they brought them to the United States, they would die. They would die very quickly over and over again when settlers brought them over because of a disease called the Eastern filbert blight. It's a fungus, and it's very nasty. And essentially, no one was able to grow hazelnuts for a very long time in the U.S. Until they started growing them on the West Coast in Oregon, where the disease had not yet crossed over because it was living on American hazelnuts in the Midwest and East Coast and never crossed the plains and the Rockies. So it was geographically isolated. And then we saw in the 70s, the disease got vectored over there. There was a large dieback of commercial hazelnut orchards, and then they were able to find some resistance and breed it in. And then, so what Rutgers is doing is they were bringing very large amounts of germplasm back from Turkey, Latvia, Italy, and trialing that to see if they could find resistance to Eastern filbert blight. So the program was a great introduction to all these things. I got to learn how to make crosses, which is you can see on the left-hand side here. I got to learn how to uh, propagate hazelnuts at scale and inoculate them and score them and plant them. And there on the right, where I'm standing in a field, those are actually seedlings that they're from nuts that I made the crosses on, which was really cool. This was a couple of years ago. I got to go back and see the field, and I felt like a very proud dad because I literally took the pollen and touched the flowers and collected the seeds and grew them into seedlings and planted them out. And it's really cool to see time measured like that. I learned a lot about grafting. This is a hot callus pipe for grafting hazelnuts. And I did an independent research project for my senior thesis where I was evaluating nut quality in a very large population of Turkish and Latvian trees that were brought back that had some resistance and identifying which ones might be the best candidates for crossing into other material in the program. After undergrad, I went to grad school at the University of Wisconsin. This is a bit of a shift. I went to a carrot, onion, and beet breeding program. My rationale for that was that I had worked with the trees and I loved them, but one of the perpetual issues in tree breeding is that they seem to be a little bit behind. The generation time is very long, so progress is typically slow, and they don't always work with the most modern methods and techniques. This was an opportunity to see how a faster paced breeding program, like a vegetable breeding program, could take advantage of new technology. And the project was also very interesting because it was all about breeding for flavor and eating quality. So essentially, we were growing these beets, and there's a few key compounds that impact whether consumers like them or not. And what we were doing was, or what I was doing rather, was measuring these compounds in the beets and seeing if I could correlate those to consumer liking, and also doing some experiments to figure out if there's different ways to grow plants or beets or different, harvest times or intervals that could affect the ability of or the flavor and eating quality. So I got to use some very expensive high-tech machinery to measure the dirt flavor in beets, which is called geosmin, and that's the same compound that makes the earthy smell after a rainstorm. Um, but I also got to see how flavor changed in beets over the course of the season. And we won't go through all the details of this experiment, but it was a really good exercise in learning how to breed for people and how to breed for what people like and how complicated that data can be. And it was also great because we got to see some sources of variation that you don't think of. For example, here, this experiment showed that the sweetness as measured by total dissolved solids, which is through a refractometer, that sweetness can fluctuate over the course of 24 hours by up to 2 Brix, which is, quite a difference. I also got to learn how to develop new tests, and that's going to come in handy for the Project Pawpaw because this was a case where COVID made one of the testing kits for oxalic acid very expensive and impossible to get. So I essentially had to figure out a new way to measure it using cheaper methods, and I was able to come up with a very interesting method, and I think it has a lot of potential. This is called a spectrophotometric assay, and that was a real task to learn how to design something like that, but very rewarding. And the coolest thing we got to do is, as I helped set this up, and then the next grad student was able to continue it and is almost through it now, is a genome-wide association study. So what we really wanted to do is figure out which genes are responsible for certain traits in beets. So we grew every beet variety that we could find in the world, and we gathered them all, grew them in one field, replicated multiple times, and then we collected DNA, little leaf tissue samples from every beet, and we sequenced the DNA, and we're trying to see if we can find the genes responsible for some of these flavor compounds, as well as shape and resistance to some diseases and all sorts of things like that. So that was all through grad school. While I was doing this work and learning more about modern plant breeding techniques and vegetable breeding and the seed industry and all the other complicated things that go behind our food system, I was also really starting to get serious about the pawpaw thing. I first saw pawpaws when I was a kid. I was 11 years old when I planted my first pawpaw. I was visiting my brother at McDaniel's Nut Grove, which is a famous nut grove at Cornell. He was working there for the summer, and I remember seeing this very tropical looking tree, and I said, "What is that?" And they told me it was a pawpaw. It's a native fruit, and it tastes like banana and mango. And I was like, what are you talking about? I know all the fruits. There's apples and oranges and like, once in a while a mango. But I was instantly hooked. That night I went back to the room I was staying in and I stayed up all night and I read every article on the internet about pawpaws, which was actually something you could do back then. And I came to breakfast the next morning and I was like, boy, do I have some fun facts for you folks. And I that was the start of it all. And this is actually a picture of me. I got to go back 2 years ago and take a picture with the very grove that started all of this. And so those are the maybe not the exact trees, but those are definitely related to the trees that started all of this. And that was really fun. One of the benefits of being in grad school was that I had access to really cool tools. This is right down the hall. Someone else was using a scanning electron microscope. So you can actually see in this picture, this is a pawpaw leaf, and you can see the individual stomata opening and closing. You can almost see intercellular structures, and this is just something fun. So that was a definite upside of being in grad school. The downside of being in grad school was that I did not have any money. So there was a period of time where I wanted to start doing this pawpaw research, and having no money, I started selling plasma and enrolling in medical experiments to get money so that I could buy the first rounds of reagents and materials for um, for this kind of work. And hopefully that presentation came back up. Um, but once I started that, I took that money and I used it to get some equipment and get some supplies. And I also started to travel around. This is sort of my first venture into actual fruit exploring, which is where I got to go down to Kentucky and Indiana, and this is the Kentucky State University Pawpaw Germplasm Collection. I got to walk around there, and I got to go to Cliff England's orchard in Kentucky and see all of his different varieties and try different varieties of pawpaws. And you can see those are shade structures from I think an Ohio State shading experiment. And I started to make these connections with people, which was very fun, and to really build the network that I need for this. I started to develop protocols. I really wanted to figure out how I could scalably process samples and breed for pawpaws. In the hazelnut lab, we were blessed because these nuts were relatively shelf stable. So as long as you could harvest them all and get them in, you could work on them over the winter. And that was what my job was. All throughout the winter, I would evaluate hazelnut kernels and take measurements on them. The challenge with pawpaws is you do not have that sort of shelf stability. You have to find a way to quickly and efficiently collect all the data you need on your fruit before it goes bad, which is a real logistical challenge. So what I started looking at was I looked at that experiment I was talking about with the GWAS, where we were looking for the genes controlling shape. Those were actually they were using a camera to analyze the shape of carrots and beets and to see which genes could be responsible for elongation and a bunch of other parameters. I was able to use that same computer program to look at the shape of pawpaws, or at least as a test, a trial run to see if it was possible, and it was. So I started figuring out how to use imagery where we could more efficiently collect data from fruit. Here you can see some test images I was doing where you can, by using that color palette there, you can actually score the flesh color and the skin color of the fruit. You can get since those are standardized sizes of squares, you can get the volume approximate of the fruit, and you can also look at shape and things like that. And then on the right hand side, you can see I started doing taste tests. So I would bring the fruit back and I'd randomize it, and I had made an online form, and I would bring people and let them try the fruit and then have them evaluate some of the flavors they were tasting, whether it was good, whether it was bad. I'd collect some basic biographical data on them as to, if they're experience with pawpaws, if they smoked, things like that, if they had just eaten, because there's so many confounding variables. So this is a lot of what I'll show you at this point are all tests. These are exercises where I could figure out how to do something, sort of get my bearings and define the program better going forward. Here's a video, if it will play, of this is analyzing texture, so fruit firmness, skin thickness, for these pawpaws, and you can see it's actually pretty quick. It's you're taking these measurements, and then they go right into you can see the picture booth there where we would take the pictures after. So that was just an example of finding ways to efficiently um, evaluate pawpaws. Let's see. I don't think something just happened on my presentation.
The video is currently well, now the video is running again.
Yeah, I'm trying to get out of the video, but I'm unable to.
Try hitting it. Try hitting
Can
escape.
can you reload?
Oh,
Yeah, you probably have to reload your presentation.
all right. Well, now I know not to click on videos.
I don't know. Google Slides can be quirky sometimes.
Yep. All right. Well, it can pick up right after it. Hopefully you can see that. We started running shelf life experiments. So I wanted to see if I could impact storage and shelf life by putting pawpaws in high ethylene environments, low ethylene environments at different temperatures. I always with controls. These were small trials, and you could see there were differences. This is a misleading graphic because on the bottom you see that the low ethylene was actually at 40 degrees, so in a refrigerator. And it turns out that temperature was the most impactful component of this. The ethylene made nearly no difference in my experiments, but the temperature was highly impactful. But this is just, once again, you can see this sort of modular environment where we're using these closed clamshells to have a bunch of treatments in close proximity with each other. It was an exercise. I started looking at seed dormancy to see if I could speed up the generation time of pawpaws by reducing their need for a cold stratification step. Essentially, the thought was that if you harvested a fruit, instead of taking the seeds out and putting them into a refrigerator in some damp peat moss for the winter, which is how you should germinate pawpaw seeds, instead of waiting all winter and then planting them in a greenhouse in the early spring, if you could find some chemical treatment to overcome this dormancy, perhaps you could get them to germinate right away, and then you could get a whole winter growing season in a greenhouse and have larger plants ready to go out in the field in the spring. Uh, there is some mixed results of that, and definitely requires more experimentation. But that's a project for when I have some more time or maybe some funding. And then along this, I also started to develop informational resources. So this is the Project Pawpaw website. This is where I started to upload planting guides and germination guides and guides on grafting. And you can see there's all sorts of information here because I wanted people to know how to grow it. I was starting to sell seedlings at that time, and I always had to explain these things. And I said, well, rather than explain them every time, how about I put them into a guide and make it open for everyone to see? And you guys can go to projectpawpaw.com/learn, and all these resources are up there, and they're continually being improved. And I also decided that I got to start putting this under some sort of brand. I got to get serious. So I got a logo made, and we built out our website, and I started designing merchandise because I knew that this work was not, in order for this to happen, I would need money. So I needed to start raising money and funds to get this experiment going. So I designed a whole bunch of stuff. You can see there's a Pawpaw Hawaiian shirts and Pawpaw t-shirts and earrings and sweatshirts. And that's all still on our website, projectpawpaw.com. And all the proceeds to this day 100% of the proceeds go directly to fund pawpaw research and breeding. I don't get paid from this. It's these just pay for materials and expenses. I started making videos because I wanted to help attract more people to my website and share the project and get the word out. So I started making TikTok and Instagram videos, and that went really well. Over the course of about a year, we got up to I think 17,000 subscribers on TikTok, and I think maybe 8,000 Instagram people. We got millions of views, literally 2 million views in some of these videos talking about plant breeding in general, my grad school research, and then eventually transitioning over to more agroforestry and fruit topics. And I would make videos updating people on the research. And so that was the point I got it to. I figured out what the project was going to be. I did some preliminary experiments to figure out the scope and the direction. I had written up a plan of what we'd do. I built a brand, made a website, started getting some publicity. I created merchandise and had it all ready to launch. And I knew that was ready to go. And that was my goal, is that by the end of grad school, by the time I graduated, I'd have this project that I could launch. But I had one thing to do before I launched the project, is an Appalachian Trail through hike. And that was something I'd been planning for a very long time. So I finished up grad school, got this all ready and put it away, and uh, sold all my stuff and got my backpack, and I started walking. Quick aside, the Appalachian Trail is a 2,200-mile footpath that goes from Georgia to Maine. It crosses through beautiful country. It's also, I mean, it was just fantastic. There's a picture of me in a flood in Vermont, which was a very, very catastrophic flood at that time. But it was really, it made me feel like there was still wilderness out there, and was very fun. Got to see breathtaking vistas. That's an alpine forest up in the Smokies. It seems to be frozen in time. There were some really big trees. Of course, plenty of pawpaws to see along the way, as well as a lot of great other flora. We got to see elderberries and butternuts and American chestnuts. There's thousands and thousands of those stump sprouts from American chestnuts, as well as some white pine blister rust in the wild. Got to encounter some animals, such as a copperhead, which bit me in Connecticut. Um, and then I recovered from my copperhead bite picking blueberries on a blueberry farm that was right along the trail. And then as we got up north, it was starting to come to an end. I'd gone through lots of shoes and met lots of fantastic people. Got spent my last few days in Maine, hiking through the 100-mile wilderness before summiting Katahdin. And then literally on the way down from Katahdin, which is the last mountain on the Appalachian Trail, that's the terminus. And this is a picture from one of the best days of my life. On the way down from that mountain, halfway down Mount Katahdin, I turned the website on for Project Pawpaw. And then it was time to go. I went off on a Pawpaw fundraising road trip all around the country, and that was the start of it. So I can give you a bit more of an introduction to after the introduction. So what is Project Pawpaw? I think we got it by now. It's crowdfunded pawpaw research and breeding. What is crowdfunding? Great question. So you may have seen things like Kickstarter or GoFundMe. Now, the approach we're doing is exactly the same. It is very similar to these. The only difference is that Kickstarter and GoFundMe both take a significant amount of money out of the fundraising for fees. So we just bypassed that and did it ourselves. We've raised over $60,000 in the past 2 years since that crowdfunding campaign went live, and all of that money has been directly invested back into our research. The money was raised both through our online store, all proceeds through that, but also through going around the country to pawpaw festivals and farmers markets and in-person events. Here's our booth that we sell pawpaw merchandise and seedlings at pawpaw festivals. Um, that first road trip was a lot of fun. We got to go all over. This is a pruned pawpaw orchard in New York, or Pennsylvania rather. We slept in a tent. We, had everything in a trailer. It was a very nomadic pawpaw existence for a bit, and that was all good because I didn't have my job yet, and I was starting my job after the fundraising season for that first year. Project Pawpaw's main goals, we got a three-phase plan. It's all about research, breeding, and market development. So the research is to understand how we can help pawpaws reach their true potential and reach the supermarket. So that's some of the things I've shown you, looking at the best ways to grow, harvest, and store the fruit, figuring out if there's ways that we can accelerate the life cycle, and that research directly feeds into the breeding program. So we're trying to make pawpaws that taste great, grow well, and are better suited for small farmers. And the research helps speed that up, helps make that more effective, helps us understand what's actually needed by the industry. These varieties, we're working to develop them. We're aiming to develop a suite of varieties, so more than one variety with different ripening times, and eventually hoping to develop ones that are better suited for different regions of the country. Because this is a decentralized organization, so we're building up a network of farms to help develop this stuff. Market development is critical because you want to make sure that farmers have a reliable path from tree to table for their fruit. So part of this is understanding how consumers interact with the fruit, understanding what consumers need, what farmers need, developing packaging, developing different ways to ship, consumer education materials, working with grocers to figure out optimal protocols, working with growers associations. So this is a really collaborative part, is the market development section. One of the biggest things we've been doing right now, and what seems to be consuming all my effort now, is getting this network of research orchards up. So we did our first research orchard this spring. It was an 800 tree orchard in southern New Jersey. And we'll talk more about that later. I think we're going to go into some specs about that. But the main goal of this is to have orchards throughout the country that we can test existing varieties in multiple environments and develop new varieties for those environments. Like I said at the start of this, one of the best parts about this project has been all the people I've gotten to meet. So here's a picture of me with Cliff England and Doug Fells, and a picture of me and the famous Neil Peterson. I mean, Neil was my childhood hero. I sent him fan mail when I was a kid. So to get to meet him, and I got to tour his orchards and talk about what he's learned and things that he wishes he had known, and get some really valuable lessons from his experience. And along with these other two gentlemen too, just understanding these people have been growing pawpaws for many, many years, and they've got great collections. And it was great to meet them, and they've become really valuable resources, along with tons of other people I've met on my journey. We've got Raleigh Pawpaw here and James Dingus. I've gotten to tour orchards all over and understand how people do things, and it's been a really lovely collaborative sharing environment so far. This is at the Ohio State Pawpaw Growers Conference. That was this spring, and got to meet even more people. And of course, just the collaborations that have sprung up through these organizations, they all sort of make a web, and it really waterfalled. So through NAFEX and NNGA, I was able to interact with all different academic partners. The North American Pawpaw Growers Association has been super supportive of the work, and I always love talking to them. Kentucky State were just a fantastic resource, and we're good friends. And I think one of the things I'd like to touch upon is that we aren't trying to replace anyone. There is so much progress to be made with pawpaws that by working together, the synergy that we have with all these different collaborator institutions has enabled everyone, and it will enable everyone to go much further than we could on our own. So Kentucky State has great germplasm for Kentucky and for a lot of other places. But we're going to be able to test that germplasm in different environments. We're going to be able to develop genetic tools and new protocols to make it more efficient. And I think everything is lining up for a really exciting time in the world of pawpaws. So now I'd like to give a brief overview of the actual breeding program, some of the objectives we have, some of the goals we're trying to work towards, and some of the tools we're trying to develop. So this is traditional plant breeding. It's the age-old tactic of using a very highly scientific paintbrush to move pollen from the flower of one tree to the flower of another. You make your controlled cross pollinations between two trees that are of interest. So if you have one tree that has really thick skin and one tree that has really firm flesh, you can take pollen from one tree and put it on the flower of the other, let that grow into a fruit, harvest those seeds, and then grow them out. This is just the protocol we use for making crosses. It's you make your pollination, then you put the flower in a bag. I like to use these cups here because you can collect a lot of pollen, and the cap keeps it from blowing in the wind. And then you grow them out into seedlings, and that's the basis of plant breeding. It's the same thing they do for every other plant, every other crop, apples and pears and peaches and vegetables, corn, soybeans. It's all done at the base level with that same traditional plant breeding, which is making a cross, so creating variation and then selecting through it. And it's not just one cross you make. You make many crosses, and you're generating hundreds and thousands of plants. The general rule of thumb, which is not a scientific rule of thumb, but typically they say for every trait you're selecting for, you'll need about 1,000 trees to effectively find one that has the combination of traits you're looking for. So you can see the numbers really quickly start to add up, which is why we need to develop new tools to maybe make it more efficient, especially if we're trying to do this in one lifetime, whereas a lot of these other crops that are more domesticated have been being bred for thousands of years, which is a fantastic feat of human culture. There's so much variation. You can see there's variation on a tree, but there's also variation between trees. So those small ones were all taken from a tree where every fruit was small like that, and those big ones were taken from a tree where all the fruit was large like that. And you can see just the difference in size. And you also have to realize that a lot of our selected pawpaw varieties are only 1 or 2 generations away from wild. I should maybe go back, and I hope that by 30 minutes in this presentation, it's not too late for me to introduce what a pawpaw is. So the
Go
pawpaw is
for it.
North America's largest native fruit, and I somehow have missed introducing that. So it's a native fruit to North America. It tastes like banana and mango, and it has the texture of a ripe avocado. It was a very culturally significant fruit. It fed indigenous people for thousands of years before people were settling in this area. And then after white settlers came, it played an important role in the frontier and settlers. It fed the Lewis and Clark expedition when they ran out of food. It was George Washington's favorite fruit. There are folk songs about it and plenty of anecdotal stories all about it. And it seems to have been all but lost as the food system got bigger and more efficient, and a priority was placed on stores being able to keep one type of fruit in stock year round. Apples made the cut, and pawpaws did not. So pawpaws have a very short shelf life, and that's their main limitation. They are a soft fruit. They bruise easily. They can go bad in as quick as a few days. And with refrigeration, you can eke that out to a few weeks, maybe a month, if you pick it at just the right time. But these challenges have really led to a loss of popularity in the commercial food system. But luckily, we've been seeing a total resurgent, and maybe a pawpaw renaissance is the right word, as people rediscover this fantastic fruit. It's very tasty and tropical tasting, and just a very charismatic cute looking fruit. And as people sort of discover this and their priorities shift towards more localized food systems and towards smaller agriculture, more sustainable fruit and food systems, I think we are seeing a really cool resurgence in love for the pawpaw. So sorry to everyone who doesn't know what a pawpaw was while I've been talking about them for so long. You can see here, once again, this is a picture of just some of the variation present out there. We've got all sorts of shapes and sizes. We're focusing on things like shape and size, of course, but also seed to flesh ratio. You can see there, the inside of the pawpaw, it has yellow flesh, and it has a series of large black seeds that are in there. And that seed to flesh ratio is something we're very interested in because we obviously would like to have more edible parts of the pawpaw. You don't eat the seeds. So having a higher or a lower seed to flesh ratio means that there's more edible flesh per pawpaw. And of course, size is important too. Flavor is critical. There is a lot of variation in flavor too. Some of the wild ones can taste like the best pawpaw you've ever had, and some of them can taste like burnt caramel or really not taste very good, have a chemical taste. So we are making sure and being very careful to make sure that the varieties we're developing have an excellent flavor and eating quality. We're also looking at skin thickness and durability. So we talked about some of the concerns uh, with pawpaws and how they don't store very well, they're hard to ship, and a lot of that can be fixed at least in part by breeding. So we've been able to find pawpaws that have thicker skin. We're hoping to find ones with, or we have found ones with firm flesh, and we're trying to cross those together. Uh, those are the main shelf life things we're looking at right now. We're looking at acetogenins. We don't have time to dive into the full acetogenin story, but essentially there's a class of compounds in pawpaws, but also in all the Annona families. That's cherry moya, custard apples, soursop. Those are all tropical fruits that have been eaten for a very long time. So these annonaceous acetogenins they have negative health effects, or at least they've been implicated to have negative health effects. And while the evidence has not been overwhelmingly strong in that it's an issue in pawpaws, and there's a few other factors such as not being able to really eat enough pawpaw to have long-term health effects. The fact of the matter is people are worried about it, and we should be selecting for pawpaws with low or no acetogenin. One of the main things that's been inhibiting that is just the cost to measure these. So it's currently measured using liquid chromatography or HPLC, or sometimes gas GCMS, which is the same machine I was using to measure geosmin in beets, that dirt flavor. So it's very expensive to measure it. And for plant breeding, like we said, you need to have thousands of plants. You need to collect the data points. So collecting multiple measurements of acetogenins in thousands of pawpaw trees when the sample can cost $100 quickly gets beyond the budgets of our t-shirts and seedling sales. So we're looking to develop new tests that are more affordable and lower cost to measure the acetogenic content in pawpaw pulp. And that brings me back to that oxalic acid assay that I had developed in grad school, which was a low cost and effective method for measuring a chemical. So hopefully we can find more funding and more support, or we can start to work on that because we think that'll be really important for helping us to breed lower acetogenin content in pawpaw. Polyphenol oxidase is another trait we're looking at. That's the enzyme that makes your apple turn brown after you cut it and let it oxidize. It's in pawpaw fruit too. While it's a bit of a ripeness indicator, because you can see that an overripe pawpaw will turn brown like that, there is some variation in it, and we will see that this polyphenol oxidase also has a impact on bruising. So little scuffs and bruises, and even the vibrations of pawpaw fruit against itself in transit can cause it to have these brown marks, which are unsightly, and the American consumer does not like brown splotches in their fruit. So we're looking for variation in polyphenol oxidase activity in the skin of the pawpaw so that we don't have easily bruised fruit. There is a lot of variation in the color of the pawpaw to begin with outside of polyphenol oxidase. And you can see these are 3 ripe pawpaws, all with completely different hues. And we call this trait the color break. So essentially, there's some hope that if you found a stable color break in pawpaws that always seemed to turn yellow when they were ripe, it could be an easier way for orchard owners to help workers pick the fruit. Right now, one of the factors impacting shelf life is the way it's being harvested. Pawpaws are only good when they're perfectly ripe. If you pick them underripe, it's very difficult to pick them at an underripe stage where they will harvest or will ripen off the tree. The flavor is far better when they ripen on their own and when they're picked ripe. So the two schools of thought are currently people either let them fall to the ground or they go and they squeeze the fruit and pick it right when it's ripe. So the downsides of letting it fall on the ground include uh, having a very soft fruit fall 4 feet and hit the ground and get bruised, which automatically reduces shelf life. And there's also some legal complications that in some states you can't sell fruit that's been on the ground just for food safety laws. So the alternative, which is squeezing fruit, also is not the best option because when you squeeze the fruit, it's time intensive, it's expensive, but it also once again bruises the fruit if you have to squeeze it. So if a stable color break could be found, we could essentially tell workers to pick the yellow fruits. Now, this has been more challenging than we had initially thought because the color break is not stable. Often it is interacting with how much sunlight the fruit gets, sort of like the blush on an apple. But we do think that with consistent pruning, we might be able to develop a system where pawpaws could have a color break as a ripeness indicator. But more work is required. And then, of course, we're very concerned with agronomic traits. Anything we release has to be not only fantastic fruit that maybe lasts longer and ships well, but definitely tastes great. But it also has to grow well because farmers need to make money. The tree needs to be healthy and robust. It needs to be resistant to diseases. It needs to be vigorous and high producing, and it needs to be adapted for the environment it's in. You can't have very late bearing variety in Wisconsin where the freeze will come before the fruit is ripe. So we need to make sure we're breeding for the context of the environment and breeding trees that are well suited for that environment. Traditional phenotypic selection is what I've been describing. It's where you go through and you measure a certain trait in a population of plants, and then you pick the best ones, or maybe you pick two ones with different traits you'd like to combine, and you cross them across. That works really well for shorter cycle crops. Here's a picture of our currant breeding field at the Savannah Institute. Currants will bear fruit 1 to 2 years after they are planted. So it's really easy to see the results of your work if you make your cross and say, okay, that worked. These plants have the combination of traits we're looking for. Because every time you get to do that and make a new selection, that's another cycle of plant breeding. It also works well for currants because currants are really small. You can pack them in tight together, and more plants equal more opportunity for gain because, if you're trying to hit those large population sizes to find the plant that has all the traits you're looking for, packing them in there per acre is really a much more efficient way. Unfortunately, pawpaws are neither a short cycle crop, nor can they be packed in at nearly the density of currants. They're taking 5 to 7 years to fruit from seed, and they do require space. Even at a super tight spacing, like the breeding orchards we planted, which are at 5 foot in row spacing, they still are not that many trees per acre, and we'd need a lot more land and management to make that work. I hope you're all ready to get your graduate degree in plant breeding because in the next 10 minutes, we're going to go through the breeder's equation. So R is the genetic gain over time. Let's think about a straightforward trait like the weight of a pawpaw. So say that I want a heavier pawpaw. In some imaginary world, that's the only thing I'm worried about is the weight of a pawpaw. So if you were to be breeding for weight, you'd be picking the heaviest pawpaws you have in your orchard and you'd be pollinating them with your scientific paintbrush and collecting those seeds and growing those seedlings and planting those seedlings out and seeing which one of those seedlings from that cross-pollination have the heaviest fruit. And then you'd continue that and you'd do that multiple times. And the rate of genetic gain is, how many grams does your pawpaw fruit increase by over X amount of time. So that's impacted by how harshly you're selecting. So if you're really lax and you're just picking, the top 1/3 of your pawpaws, so you got some really heavy ones, but there's some, intermediate heavy ones, that's going to reduce the efficiency of your program. Your selection accuracy is also important there. So maybe you have a bunch of undergrads that are weighing your pawpaw fruits, and they're maybe not doing a great job. So you have some really tiny pawpaws accidentally getting mixed into the trees that you've selected. That would slow down your progress. You have genetic variance, so that's you need to have variation to make gains. So if all of your pawpaws in the entire orchard were all the exact same size, it wouldn't be possible to really make any gain over time because there's no biggest ones to select from. You need variation. So luckily, there's lots of variation in pawpaws for all these traits. So you'd be able to go and find your heaviest pawpaw in that uh, orchard and then cross those together. And then years per cycle. That's all in the context of no matter how good you do all these other things, if it takes 5 years to go from seed to seed, at the end of the day, you make your pollination and then you won't know if it worked or how much progress you made for 5 years after you plant those seedlings. So we're looking at that years per cycle thing. I mentioned before we were talking about speeding it up. So life cycle optimization. Uh, this is a picture of currants that I have in a growth chamber for my full-time job at Savannah Institute, but it's things like this where you can manipulate light levels and fertility and temperature and things like that to try and get the plants to grow really fast. And maybe you can get them to flower in 4 years instead of 5 years. It's things like dormancy induction, where if I did grow my plants in the winter, I'd want to make sure that I could plant them when they're dormant, or because these plants do need a winter period. They need to have winter. They're deciduous plants, despite the fact that they look very tropical. They need a winter. So if you're going to be doing these augmented life cycles in a greenhouse, you need a way to make them go to sleep for the winter. So figuring out how to put them in the dormancy, then you store them in a walk-in cooler. The seed stratification experiments, like I was talking about, trying to skip that 4 month seed stratification would increase your speed by 4 months. And that's what they do in these big plant breeding programs is it's all about shaving off weeks or months. And if you do a bunch of these techniques together, you can actually start to make some really significant decreases in the generation time. And of course, top working your seedlings onto established trees. So after I took my, or I'll talk about that in a second, there's that speed breeding again. That's the seeds being soaked in different seed treatments, and you can see that they get a little weird after some of the treatments, like the gibberellic acid makes them kind of contorted. So there's some dialing in to be done there. This is top working, so it's grafting. So essentially, you could grow your seedling from the seed you collected after your cross, and then you could cut the top of that seedling off, and you could graft it onto a established tree. And consistently doing this, I've been able to go from a scion that is 3 or 4 inches in length to something that is 3 foot in length after just the first season, which is much faster than a seedling tree would grow on its own roots. And this is one way, if we were able to get orchards of established trees, we could test our varieties even faster. That would really be helpful. Without going too far into detail here, this is just an example of what some of these different techniques could do to speed up the process. In traditional, you would collect your seed in the fall and you'd stratify it over the winter and you'd plant it in the spring and germinate it and you'd let it go dormant and you'd plant it out in the field the next year. Then you'd wait for 5 more years until it flowered and you could evaluate the fruit. In a top working system, you might collect your seed and stratify it over the winter and then germinate it in the spring, grow it for 1 year, and then once it goes dormant the next winter, you could take that top off. You could take a scion from it and graft it onto a mature tree, and then it would flower a little earlier. And then idealized hypothetical scenario where the seed dormancy skipping works, you could get your seed in the fall from the fruit, and then you could treat it with a chemical and then instantly germinate it over the winter, grow it in a greenhouse, induce dormancy and put it into a walk-in cooler, take that scion off and graft it in the summer to a mature tree, and then hopefully it would flower in a couple of years after that grafting. So that would be a wonderful scenario, which I think we're still a little bit away from, but it's a good goal to have. Given time, I think we'll shorten your master's degree in plant breeding just a little bit. So the selection accuracy is something else you can modulate, as well as selection intensity. So if it's all about population size and you're trying to get as many trees as you can, a more efficient way to do that would be to be able to screen the trees and only plant the best ones. So if you had space for 1,000 trees, instead of making your crosses and growing 1,000 seeds into 1,000 trees and planting them and hoping that you have the best tree there, you could instead screen those trees at a seedling level and plant maybe just the top 50%. So you knew that at least if you grew 10,000 seedlings and then picked the best 1,000 out of that, you'd have a much better chance of finding the next best pawpaw variety because you'd be you know enriching your assay. So that's called genomic selection. We're going to go quick through this, but essentially it's using information from DNA to predict traits in seedlings. So it's looking at the genes that control certain traits, and you're looking at the DNA of it. You can identify your promising individuals earlier, such as when they're in these little crates, instead of planting them out over acres. This many trees can populate 1 full acre of trees. But if you could do this and screen them at this stage, it's much cheaper. You don't have to pay for all that time of mowing and maintaining the land use and just the time spent maintaining these trees in order to see if they make good fruit. You could just predict which ones make the best fruit and then spend that time and money growing those trees as best as you can. This is DNA. We really are diving into it here. It's made up of a bunch of little bits called nucleotides, and what you can essentially do is you can you can you can look at little fingerprints all around the genome. So humans have this too. If you've done like 23andMe, that's what they're doing. They're looking at lots of, maybe 200,000 little fingerprints, snippets of your DNA. There's billions of base pairs in your DNA, but just looking at a snapshot of them and seeing evenly spaced fingerprints, and then seeing if you can correlate those fingerprints to a specific trait. This is an example we did with chestnuts, where you can use imagery to look at how many chestnut burrs are on a plant. Then you do that a lot, like with many, many plants, like a lot of plants, then you could correlate that to essentially your DNA sequencing. And what that does is, whereas your typical traditional plant breeding is you make your cross, you grow your plants, you measure your traits after they're mature, and then you select parents and you continue. With genomic selection, you make your cross, you could grow your plants, and then you could select the ones that you want while they're still seedlings, then maybe graft those onto big trees and get them to flower really quick and make your cross. And it dramatically increases the speed with which you can do these generations. So this is a complicated, convoluted graph of that. So what we're trying to do is with this research orchard is we're trying to set ourselves up for that. We're still very far away from genomic selection, but it is absolutely the future. It's already the reality in crops, like in vegetable crops and row crops. It's becoming a reality in more established tree crops like apples and peaches. And it's certainly the way to go with pawpaws. It's still traditional plant breeding. It's not genetic modification. It's the same thing it's always been done. It's combining pollen from two plants, except it's just more efficiently picking out the winners from those seedling populations so you can focus on them. So to do that, we needed to get plants in the ground. That was the issue. We needed to get going. So this is my best friend David Lubick. He went to grad school for plant breeding and genetics. We were roommates at Rutgers, and we worked in the hazelnut lab together. And he also happens to have a family vegetable farm in southern New Jersey. So he stepped into Project Pawpaw as our research farm manager. And this spring, we planted a big seedling orchard down there. You could see it was here's the site prep. It was used for intensive vegetable production for 40 years before this. This is a sandy loam soil. It's very well drained. It was in a cover crop of ryegrass, which was terminated, and we disked it in, and we used a vegetable plastic layer to lay woven ground cloth. That's kind of like a landscape fabric mulch to keep the weeds down in the row. This is what it looked like after we laid all the fabric before we planted trees. There's David. I built this tool that allows us to mark out 5-foot intervals. So that is a little sponge there with some white paint soaked into it, and you run it down the row, and it makes a dot. I did the math, and every time that hits is 5 feet between the plants. Then we use a modified weed torch with a coffee can to burn perfect holes in the fabric that we can plant into. Went through with a drill-powered auger, and that's how we dug our holes, which was really quick and efficient. Although I'd suggest you get a longer auger so you're not hunched over all day, like you can see my friend Ian is here. Then we planted our trees. You saw those crates before they were planted. We let them go dormant, and then we planted in April. We separated them out into sort of a bare root and put them in a bucket with water and some mycorrhizal inoculant. That was what we planted. So we took those buckets around and we planted them in each hole. We each plant, after it was planted, got a fiberglass tomato stake, and we've also been using these paperboard tree protectors. So instead of plastic tree tubes, these are 94% paper and 6% very thin plastic film. Since we knew these were going to be single use in this location, we put these out there, and we did a test plot before. We can talk about pawpaw establishment later, maybe in the Q&A, but we did a preliminary row before we did this whole big planting where we tested a bunch of different establishment procedures, and we saw that tree tubes dramatically improve establishment, and that these paper tree tubes were good enough to get a tree well established. They gave it protection from wind, they kept the direct sun from scorching the leaves, and they helped it a little bit as it was getting established. And we saw dramatically better growth in pawpaw trees that were tubed versus pawpaw trees that were under shade structures that were made out of cloth and fabric, or versus controls. This orchard is also irrigated. There's drip tube underneath that weed fabric that allows us to keep it watered in this sandy soil. So yeah, it was a lot of fun. We got it planted over the course of about 5 days. It was a lot of work. My lovely family came down and helped me. That's my girlfriend Ella. Uh, she is the main victim of Project Pawpaw, who gets cajoled into coming onto pawpaw road trips and working at pawpaw festivals and planting pawpaw orchards. And we had a whole great team. You can learn a lot more about this at projectpawpaw.com/orchards. You can find all the specs with the inter-row spacing, in-row spacing, what we use as a ground cover. There's a 20-minute long full-length video on us planting this orchard that I implore you to go check out. Real quick, in the last bit here, it's the time for the past, present, and future of the pawpaw industry. I should just go back here and say this is just the first of these orchards. We're going to be planting these all around the country, hoping to do one in Wisconsin. We might be doing one in Georgia coming up. So the goal is to use these as training populations for that genomic selection thing I was telling you about, where we can predict the seedlings' value when they're little seedlings. But first, we got to get one generation of orchards fully grown. That way we can use that data to make the algorithm to predict it. Past, present, and future of the pawpaw industry. Real quick, people have always loved pawpaws since they first come in contact with them. They've been enamored with them. I got the chance to go and visit the Rare Manuscripts Library at the UIUC, University of Illinois Urbana-Champaign, and they had original Audubon prints. That's an Audubon original print of pawpaws from hundreds of years ago, and these were all original plants. And that's JP, who was really kind and reached out to me to show me all around, and it was just a fantastic day, and just goes to show that people have been into these for a long time, even though we've kind of forgotten about them now. But we're not the only ones that are into them. For a long time there are these niche fruit that people didn't know about. Here's the number of pawpaw festivals in the country. You can see an obvious trend with an explosion of pawpaw festivals this year. There was one for many, many years when the Ohio Pawpaw Festival was the original one. And you can see this is a map of the festivals across the country today. They are all over the landscape, which is really great. People are starting to rediscover their local food shed, and this is a great opportunity to overcome those issues with shelf life, where pawpaw farmers and growers and foragers can sell their fruit at a festival right there and not have to worry about shipping it. And the people come to the fruit, which is a really interesting thing. Just an explosion of popularity too. These are all, Project Pawpaw itself has been featured in the Washington Post, The Guardian, on NPR, in Civil Eats, and a few magazines and some podcasts just this year. And this isn't even all the appearances we've had. And it seems like every year there's more and more articles about pawpaws, and people are just getting really excited about them. But it's not just Americans getting excited about them. A couple of weeks ago, I got to visit Croatia, where there's a number of people growing pawpaws, uh, which is, very far from here. It's by Italy. It's across Croatia and Slovenia. Both have burgeoning pawpaw industries with large numbers of trees being planted out. These in Croatia, the area we were is about equivalent to USDA Zone 9, although some of the sites were Zone 7 across the region. Here's an
Hey,
example
Adam.
of, yeah.
We're a little bit past the presentation time. I do
Oh,
want to include some time for questions,
yeah.
but this is good content. I think this is really great content actually. And so I know you're probably pretty close to the end, but I'm just letting you know.
Okay. Yeah, I'm right in the home stretch. We'll do quick. These guys have a 5,000 tree pawpaw orchard, which is bigger than I think anything we have in the U.S. It's just a few years old and they're growing like mad. Here's some more pictures of those. It's just beautiful out there. This is an older pawpaw planting, about 15 years. This is Ivan Sulag, who's been promoting and pushing pawpaws for a long time and has formed sort of a cooperative out there. And he's got about 30 hectares, which is about 60 acres of pawpaws all across the country, with different growers growing fruit, and he collects and markets it together. They they make and sell processed goods like I have some here like pawpaw champagne and pawpaw beer and pawpaw honey and pawpaw marmalade. But they also have their own issues. They have pests and diseases that we don't see here, such as these boring insects that make the tree break in half, things like that. So we could use that as inspiration for people like the North American Pawpaw Growers Association, which if you grow pawpaws, you should definitely join. And we can start to work on some of these issues and learn from what other people have learned or other people have figured out and start to make a stronger industry. And we need you. So if you find an outstanding pawpaw that has some interesting trait, even if it's not a great trait, even if it's just a very notable trait, for those sort of genetic studies we're working on, that's what we need. And we'd love for you to get in touch and maybe we can get some scion from it or some seed. Um, and of course, if you feel like supporting our work, all the proceeds directly support pawpaw research and breeding. It's all things that are for sale on our store, but it's also, if you don't feel like buying anything, you can sponsor trees. You can set up a recurring sponsorship every year. Even $10 can get a tree in a pawpaw orchard. And at this point, we've put thousands of hours unpaid into this project, and uh, we just really need the support to keep the work going. So with that, I will turn it over to questions. And sorry for running over, folks.
It's okay. I think Adam, there's not a lot of people that talk about plant breeding and the importance of plant breeding, and have done it in an illustrative way that opens up the kind of takes back the veil of how to do plant breeding. And as you highlighted, the principles of plant breeding extend very well from species to species, and I personally really appreciate it. Thank you. Um, so we do have a number of questions. I'm not going to go in order, but there was Eric Bina asked a question about the Sam Norris accessions, and Sam Norris did culture scene breeding to create
Hmm.
putatively tetraploid potato, or tetraploid pawpaws. And I work with potatoes, so I said
Gotcha.
potatoes for some reason. But anyway, my understanding is there's only Sam Norris 5 and 7, and I know Lehman used that material in his breeding work. I haven't seen any information really on ploidy, and I think that it's kind of out if ploidy matters that much in pawpaw. It seems to maybe increase the fruit size, but I don't know. Do you have any thoughts on ploidy?
Yeah, so the goal of the ploidy initially was to try and create a seedless pawpaw. It's the same way that
Right.
seedless watermelons are made. You double the chromosomes in one, and then you cross that with a regular diploid, and then you get something with three sets of chromosomes that, in theory, makes seeds that don't develop. That's how watermelons work. But it's never really that simple because to make it work in watermelons, they use thousands and thousands, or at least hundreds of test crosses to find a combination that actually makes it seedless. And I think that's what happened with pawpaws is that there were just a limited number of these te putative tetraploids developed, and they were, crossed with diploids, and the offspring had seeds. So I do think that has some promise, and NC State has been had a little simmering project for a very long time where they've been creating tetraploids with colchicine and trying to cross them. They still haven't had luck with it, but I think if we scale up, there's some potential there. And also the tetraploids sometimes they appear. I mean, no one's. I don't know if we've done the flow cytometry on all of them, but I think the tetraploid candidates typically have larger fruits. I think it would be interesting to mess with the ploidy overall. It does make it more difficult for breeding down the road if you're going to try and do the breeding I've described on complex polyploids. It quickly gets to be a mess. So maybe it's a matter of taking the best candidates that you have and then seeing what happens when you treat them with colchicine or isolin or something like that.
Mm-hmm. Uh, I have another question. So, and this one's actually about the fragility of roots. And so Mike N asks, by what mechanism are pawpaw roots fragile? How do you ship pawpaw bare root? And I think he's meaning pawpaw seedlings.
Yeah, we don't ship bare root. I when we sell seedlings, we ship potted seedlings. Although I've planted hundreds of bare root seedlings too, and it really they are fine. They're vigorous. You have to take care of them like any other bare root plant. Don't let the roots dry out. Uh, Plant them when they're dormant is a big thing. Um, yeah, it's I think the bare roots aren't as good for the home grower because typically a potted plant is a bit more robust, and I think home growers have better success with it, which is why we're selling potted plants because those plants typically end up with the home grower.
George Zhang asks, I think, an important question, and I would like your opinion on it. So pawpaws are the genera is kind of small. There are some species that can cross, and he's asking about crossability of species. Have you worked with Parviflora or Asimina x piedmontana? I'm just going to add that on to his
Yeah.
question.
I've got a few accessions that are interspecific hybrids. It's Yeah, I think it's definitely interesting if you're looking at things like dwarfing, or I know that some of the people in northern Florida or even central Florida are hoping to get better fruit quality into like a parviflora or something that has the heat tolerance or salinity tolerance. So I think it's definitely an interesting area of exploration, and interspecific hybrids have made a lot of the species that we eat today, like strawberries, are the classic example of interspecific hybrids making something awesome. Once again, it's a numbers game, and I think that we'll just need to put a more concerted effort into it and generate more seedlings to test them out.
I have a little Parviflora that has the fruits are really small, and that's one downside of Parviflora. But it does have some of those acetogen and bitters, but it has bright green flesh, and it has a weirdest,
Hmm.
most awesome flavor. Um, but also, Matt Stevens, one of our board members, was asking, "Do you have any research orchards in Canada?" And he says, "Would you want one?" And then he also says, just as another aside, he said, don't forget the huge country immediately north of the U.S. There are pawpaw festivals and lots of pawpaws growing up here too."
That's totally true. That's on me. I neglected to include that stuff. But yeah, there's like the Canadian pawpaw growers Facebook page and stuff. And I think the we don't have anything in Canada, and I hesitate because of just the technical and logistical issues of getting plant material up and back from there. Although I will say that Wisconsin is just a stone's throw, and I think you could it could be a good proxy for some environments there.
Hmm. Hansel de Sousa, if I apologize if I'm mispronouncing your name a little bit, but he asked about the Holy Grail. Everybody asks, can you tissue culture pawpaws? And I know Arnold Peterson is working on some of that, and some other people are. Do you have any insight into that?
I think, well, there's two questions. It's can you, and almost definitely you can probably tissue culture everything, and it's can I? I cannot. And that's the issue, is that it's very complicated, and even once a protocol is developed, even moving it between facilities often causes it to have to be completely redeveloped. So I do have good faith that in the coming years we're gonna, people are gonna get it. There's now the economic incentive because people like these plants and they're becoming so much more popular that the market's gonna drive someone to figure out how to do it tissue culture, and it's just gonna incredibly benefit the research side of things.
Um, I wanted to say that I found it interesting that nobody has raged on about acetogenins and pawpaw. And as an aside, you probably know this, but avocados also have acetogenins, and nobody ever complains about avocados. So,
Yeah, don't get me started. I had to temper myself because we could probably fill up a 45 minute slot with a talk on acetogenins. So
yeah. Now somebody was asking, let's see here. Juan Carlos was asking about, have you studied the ones growing in Japan? I know Japan, Korea, and China are there are separate breeding efforts. Are you in contact with any folks over there?
I'd love to be. I have not gone yet. Yeah, I'm highly interested, and if anyone has good contacts, please share them. Aside from my Facebook lurking, I've not gotten to interact with them yet.
And then Joseph asks, what were the cultivars that are being grown in Croatia? Was it a bunch of cliff stuff?
Yeah, I think there's about 10 cultivars in that are being distributed. There's the Peterson stuff is not there, but there's they were growing a lot of sunflower and Overlease, I think. I have it written down somewhere. But yeah, each that 5,000 tree orchard, each single row was a variety. Obviously, they were repeated throughout the orchard, but it's all grafted stuff there. Although the 30-hectare one is a lot of those are seedlings, so kind of two different approaches.
Cat B says, "Hi, I'm working on starting a pawpaw nursery. Any suggestions for early growing slash establishing trees for the purpose of scion collection? All resources or literature." I think they're trying to say all resources or literature are welcome. I'm finding out uh, something about trying to go for highest fruit yield too. I don't know. It's a little bit garbled.
Yeah, I mean that's the right? Like everyone wants to grow their trees fast and have lots of fruit. Uh, Full sun, uh, fertile soil. Yeah, I would caution against buying like one of those 5 foot trees in a burlap root ball for $400 because it's just going to be stunted while it puts its roots back out. So I'd say focus on a plant with a really healthy root system and um, you know, adequate and frequent fertilizer for the first half of the summer, then stop midsummer so that they have time to harden off. And that'll help irrigation too.
Uh, and then do you this is just a question of mine that I think people might want to hear, but do you have any insight into fertility requirements for pawpaw?
I don't. It's a something that needs to be worked on more. I know that's an interest of Kentucky State as well, and it's a lot of people have been calling for it. Someone who really does an excellent job. There's so James Dingus. He is able to. He's a guy who runs Pawpaw Chronicles, and I've seen him grow seedlings 4 feet tall in one season with adequately managing fertility. And then also there's a think Jim Davis, that pawpaw orchard, they do foliar testing of their leaves in the growing season to determine their nutrient input. So I think uh, it'd be worth having a better and more cohesive chat with them to figure out some good recommendations. And Kentucky State does have recommendations listed, I believe.
Do you have any issue putting pawpaws on fabric and seeing rodents girdle the roots or the stem?
We haven't yet, although there's going to be rodent control in the orchard. A big thing is the type of ground cover you use and not having too many legumes in the ground cover because that can serve as excellent food and hosting area for the rodents. I've also heard anecdotally that if you leave 1 foot swath in the middle of each row unmowed, then the rodents want to go live in the grass versus burrowing under your fabric in the winter. But I have not tested that yet.
Matt Soltis, I believe. He says, regarding pawpaw establishment and your experience with tree tubes, have you found full sun exposure to young seedlings is actually a detriment?
Uh, it it super depends on things, right? So if the seedlings leaf out in full sun, they can typically take it. You just have to be really prepared to keep them well watered and take good care of them. Uh, this is a windy site, so we found those tubes helped keep the little seedlings from getting whipped around a little bit. They also just offer a little bit more visibility to farm workers who, varying levels of skill. And yeah, so we take them off after the first year, essentially.
Mhm. Uh, and Curtis Reynolds actually asked a persimmon question. He says, is there any publicly available information accessible for the cold hardy persimmon trials that are undergoing at Savannah?
Yeah, go check out SavannahInstitute.org. Once again, it's Savannah, SAVANNA, no H, .org. And there should be a fact sheet on the breeding program there. But yeah, it's essentially death sentence to lots of we have lots of them out there and seeing which ones can survive the cold. But these mild winters have been messing with things. We had a really mild winter last year and nothing died. So we're hoping for a more intense winter this year.
Um, here's a question that I haven't seen asked, but that is an interest of mine. So there's current efforts, Woody Walker, others doing the freestone pawpaws.
Mhm.
Would you be willing to speak about that at any length?
I think it's a great trait. The one on this question page, I would say, has a freestone trait. It's just the lack of that little sac around the middle of the fruit. So some of the seeds have flesh clinging to the seeds, and I find it off-putting to the consumer, and it reduces your yield during processing and stuff like that. So I'm very interested. The jury's out whether how defined of a trait it is. Is it a matter of flesh firmness? Is that flesh just more firm? Is it a matter of the stage of ripeness? Probably all of those things, but nonetheless, I think that's probably the way forward.
Hmm. Um, what about the other thing I think about too when it comes to persimmon and early golden progeny is genetic bottlenecking or keeping out of genetic bottleneck in the future. Do you have any thoughts about that?
Yeah, it's a concern. I mean, luckily we've got so much germplasm that is great out in the wild. So I think we're
Hmm.
gonna have, and we're so close to the wild stuff still that it's not a huge step back to breed with a great wild accession. Whereas if you were trying to like breed corn with a wild ancestor, it takes you 10 years of back crossing to move one trait in. Um, so that's a great news. You do have to be cautious of it. And especially once you start to get into these sort of genome assisted selection techniques, it's actually something you factor in with your selection intensity. You don't select too harshly on one trait, otherwise you will likely bottleneck your breeding program.
Hmm. Um, George Zhang says that outside of Albuquerque, New Mexico State University Cooperative Extension has some pawpaw in their research orchard. They're more than 10 years old. Unfortunately, they have rows back annually. What would be your suggestion to grow pawpaw in cold climate?
Yeah, I think I was just chatting with someone about this. Out west, I think is quite harsh because uh, with so little humidity in the air, you guys just have really quick season changes. And I used to work in New Mexico, and you'd have like 50-degree temperature fluctuations day and night. And I think that's probably part of the mechanism of cold damage because Buzz Ferber grows pawpaws in Zone 4A Vermont, and they don't die back very much. And sometimes the fruit doesn't ripen because the season's not long enough. But I think the difference there is that Vermont's moderated by a ocean. The temperature swings are not that extreme. in Wisconsin, which is zone 5A, we see pawpaw trees growing well and not freezing back in the winter. So I'm curious if it's the temperature swings in New Mexico or if it's also just maybe the plants are very stressed from being in that arid climate. I don't know. I don't know anything about their site and what type of soil or irrigation or whatever they have, but
I have a question. This has been a long debate, but what relationship do you see between color and flavor? I mean, a lot of people have a lot of different opinions. Some people really liking white flash pawpaws, others liking deeper golden. Anything you want to add to that?
yeah, so the famous quote that correlation is not causation comes to mind, um,
Yeah.
because they're without a doubt differently they're different traits genetically. Now, whether it so happens in the current germplasm varieties we have that there's a strong correlation between flavor and one color, that may be. But if you were to start to breed for flavor just based off of color, that relationship would quickly break down. Um,
Yeah.
yeah.
Hey, Chris. I just
Yeah.
have a quick question. I guess as a co-host or host, you're not allowed to answer. You're not allowed to put anything in the Q&A. So, Adam, I'm going to ask you directly. So, around here in Kentucky, a few friends of mine, we've thrown around the idea of, or the question of like, why aren't there some pawpaw rootstocks? Because the rootstock, when we're grafting, can have a profound effect on the resulting fruit down the road. Some can be large, some can be undersized. It's very variable. So it seems as if, I mean, this burgeoning industry or fruit or sector of the fruit world needs some type of standard rootstock to get the most out of the pawpaw itself or that variety or cultivar. So, are you looking with all of these thousands of seedlings that you plant? Are you looking for any type of traits that might, like down the road, to you say, okay, this might be a nice rootstock to work with that sizes up the fruit well, that we could probably propagate out and offer as a rootstock?
So it's nice. It's actually an answer to your question as to why there's no rootstock. It's because there's not a feasible way to propagate it right now. So you'd need to either be able to tissue culture it, or you need to be able to do it in some sort of root layering system, which I think is feasible, but no one's done it. No one's, doing a mound layering system and making pawpaw rootstocks like that clonally. Uh, and there have been various efforts to breed for a rootstock. Kentucky State has notably done it they've tried it over and over again, and there there likely is some variation. The experiment itself is really hard if it's unreplicated because you can't clone your stuff very well. Um, I think it's going to be very important. That's why people were looking at the interspecific hybrids for a dwarfing sort of capacity. But I think besides dwarfing, I think, like you said, a vigorous rootstock could be useful too. Um, and we do have plans. Hopefully, it's not our top priority just because there's so much other stuff to be done. But in a perfect world, if we had funding and access to land and that, yeah, we definitely would put a rootstock trial in where we'd plant seedlings and graft the same variety to all of them
Yeah.
with replicated checks to see the differences in rootstock effect.
Okay, thank you.
Um, and this is more praise. So Joseph Postman, the retired pear curator, a wonderful man, says, "Thank you, Adam, for one of the best talks I've heard in decades of my NAFEX membership. Accessibly presented technical information balanced with history and useful practical info." So
Thank you very much.
the
That's very
stutter's
nice to
my
hear.
own. Juan Carlos also asks, are you also interested in the uses of the leaves for medicinal purposes?
That's a whole can of worms. So I that is I'm honestly gonna say it. That's totally outside of my area of expertise. And if that was someone's area of expertise and had the capacity to do that research, I'd love to support them on it. Um, I've also seen people interested in there was interest in using the leaves as insecticide,
Hmm.
which ultimately had some viability issues with the extraction efficiency. But yeah. if there's someone with a solid plan, I'd love to support.
And most importantly, here's maybe one more for you. What is your favorite variety? I know it's a stupid question,
Ooh,
but you know,
I know I'm
all right,
gonna
everybody get ready to search it.
start to get some death threats from all the pawpaw breeders out there. I mean, Susquehanna is just a fantastic variety, and uh, so is Chappelle. And
Mhm.
Shenandoah is a nice light fruity one. From a breeder's perspective, I'm really interested in the extremes. So in addition to you can make something called a mapping population. If you have two very extremely divergent fruits, you can cross them together and grow like 200 plants, and then you can get a really good idea of where that gene lies. So like Alhorn's white pawpaw or Horn's white flesh is that's so cool because that you cross that with one of those dark fleshed Kentucky pawpaws, and you might be able to figure out where the pigment gene is. There's one I came across that I've colloquially named the grape pawpaw because it makes pawpaws that are the size of a ping pong ball and makes them in clusters, uh, which is totally useless from a breeding, as a fruit, but very useful if you're trying to figure out the genes that control the number of fruit in a cluster. Um, so yeah, if you folks are out there and you have some really wacky extreme pawpaw that even if it doesn't taste good or it's not a great candidate for some reason, it could have a lot of value.
Uh,
Uh,
Adam, I have one more question for you, and I'm going to somewhat answer this as well. Matt Stevens in Canada, for those grafting questions, you'll want to look back at one of the interest groups that we had Dr. Pomper on from Kentucky State University talking about which grafting methods are the most successful down there in Kentucky for them at KSU. He went over that in the PowerPoint, so please access that online. But I would also like to give Adam the opportunity to ask you, answer the question. I think what Matt was asking was, do you prefer cleft grafting, bud grafting, or whip and tongue? And then also he had asked a question about dormant grafting versus actively growing.
Yeah, I love Cliff England's modified splice graft is my favorite graft ever. I put one bud on there, I get really high take rates. If you go to my website, projectpawpaw.com, there's a video of Cliff doing that graft on there. And I'm sure Kentucky State's shown it too because he's down in Kentucky. Uh, Graft actively growing pawpaw rootstock, get it like vigorously growing. Um, uh, yeah, because you like I graft in for top working, you can go as late as June or July, and I've had them take and top working is like a modified or it's a bark inlay graft or a cleft graft. But yeah, dormant scion, actively growing pawpaw rootstock.
Thank you.
well, Adam, we went a little bit over, but I don't think anybody really cares. I really appreciate you taking the time here to give this wonderful presentation, and on the behalf of NAFEX, thank you for coming today. Um, I wanted to acknowledge one more time our sponsors, One Green World. Use code NAFEX, all caps, to receive 10% off your order. Copenhagen Farms Nursery that produces rootstock, and Southern Cultured Orchard and Nursery, which is our fellow board member Larry, who is out of Mississippi and produces a lot of good low chill hour varieties. Once again, the recording of this session will be made available on nafex.org webpage in a couple days and posted in about a year to our YouTube channel. There is a ton of other downloadable content on our website as well, so go check that out. Don't miss the upcoming sessions today. We've got a couple more coming up, so stay tuned for that. And stay connected on our Facebook page and Instagram and all that good stuff. And you can find all those links at nafex.org. And thank you again, everybody, for joining us. Adam, do you want to say anything as last words?
Thank you folks so much for listening in. Sorry, we had to go real fast. I hope hope you are all experts in plant breeding now, and check out projectpawpaw.com and share us on Facebook and all the other stuff.
All right. Thank you, everybody. Take care.